I'd like to talk about what we're aiming towards for super resolution multi-omics, DNA, RNA, protein, et cetera, for maybe multiple mycoplasma, starting with the SYN3 type that you're all familiar with. And first, I want to show you where we're coming from, some qualifications. I didn't put it on the slide, but I started on mycoplasma at the Channing Lab at Harvard in 1973 when I was just a starting undergraduate. And then we didn't really publish anything significant until 2004, where we did the first combined genomic and proteomic mass spec in the same paper, thanks to Jake Jaffe, the first author there. And then Tony Forster and I wrote 2 papers about synthesizing in vitro systems. So this was different from synthesizing a genome. This is, and we haven't yet succeeded on this, although we published a few experimental papers as well. But that was the beginning of our thoughts of minimal genomes, which was definitely inspired by some of the things that were going on at JCVI or its predecessors. And then there's this 3-dimensional structure of a bacterial genome that Mark Umbarger led in our lab. And then this last one is not from our lab, but I think it represents some people that are on this call, including the Goodsell senior author. And that's about structural models. I'm a crystallographer by training, so I'm inspired by that. And in the upper right and below are some of these structural models that we did back in the day. But fast forward to today, things are exponential, both reading and writing, and we've from a imaging standpoint, so we're framing almost anything that you can sequence, you're doing by fluorescent imaging, and we're just taking that to the next step where we can get as good as 2 nanometer, 0.2 nanometer precision. And I'll show you that in a moment. And these are 2 instruments. Is there something? These are the 2 instruments that that our lab and the Wu lab have used. The highest resolution right now is the Bruker-Vutara, and I'll show you some data from that in a second. So this is the most extreme version. This Ming-J Dai was in our lab for both graduate school and postdoctoral work, and this is a paper that he wrote in Nature Nanotech, where he and Peng Yin, our frequent collaborator, showed this spatial resolution, or precision, I should say, of about 2 angstroms, 0.2 nanometers. This is using DNA nanostructures, so you know where everything is. Oligopaints partially goes back to our multiplex sequencing in my thesis in the upper left, in 1984, but really was accelerated by the Wu lab, showing these oligopaints where you have the black region which binds to the genome or transcriptome, and then 2 flexible hybridization tags that can be synthetic and anything you want. And so when we first published multiplex sequencing, was 42-plex, and the Wu lab is up to million-plex. And here's some of the, I'm going to do DNA, RNA, and protein in rapid succession. This is 3-dimensional DNA in situ. These are little pieces of human chromosome 19 from the maternal and paternal chromosome. And you can see you can display them very much like any other macromolecule, where you have this super resolution imaging.
George,
yeah.
George, how many base pairs are we looking at when you say a little piece?
So this is on the order of a few 100,000. I don't remember exactly for this one.
Okay.
The smallest region they've done is 5,000, and the largest is whole chromosomes. In fact, they've done multiple chromosomes, and we've done it by other methods as well. You'll see in a moment. So here's a kind of a zooming in on a particular loop. So this is one loop of the DNA. And you can see the loop closure by orange and red binding together region. These are 10 kb regions, and you can see multiple balls within the 10 kb. That's giving you some idea of the resolution here. And this is, again, from the Wu lab. And this is RNA, single cell each dot staying in place, changing color, GATC, the 4 colors, 4 bases, and then you can have the computer recall all of those rounds of fluorescent next-gen sequencing essentially in situ, and now each of those vertical lines was a barcode, a RNA barcode. So we're not getting every single RNA molecule, of course. We aspire to do that, but you'll see in a moment how we can get more real estate to do that in. One way is to get higher resolution, and the other way is to get expansion of the cell in a moment. So that's DNA, RNA. This protein, we basically reduce it to a nucleic acid problem by tagging antibodies to the proteins with a short 9-mer oligonucleotide, and then that can exchange with a fluorescent oligonucleotide. We can either do these big IgG, where you lose a little resolution because of the size of the antibody, or these smaller nanobodies, which are as small as 4 nanometers in its longest dimension. And these are great for multiple labels on a single protein, for example. We use this DNA exchange imaging, very similar to the oligopaints I was talking about earlier, to get super resolution. And then here's an example of some of the protein data where we show conventional microscopy on the left, and then exactly the same part of the same image at super resolution with this DNA exchange imaging method. And you can see the things that we couldn't tell apart. These are synapses, but it could be anything where we've got multiple proteins, 4 proteins, just to determin the directionality of the synapse, and then another few that determine whether it's inhibitory or excitatory. So you can learn a lot about, and it matters. This is subcellular resolution, and you just can't get this by treating the neuron as a sphere or taking the nucleus out of the neuron and hoping to learn. This is connectomics, and I think it's important. So then we have collaborations with Ed Boyden and Peng Yin. I've mentioned Peng a few times already. Ed is quite interested in this project with mycoplasma, and he and I have collaborated on DNA, RNA, and protein in a different way than the ones I've described so far, where typically there's expansion in the case of, well, in all these cases, but the Paint et al. Paper was mainly a way of sequencing de novo by a combination of in situ and ex situ. And this is some of the data from that, showing all the chromosomes, maternal and paternal, in a zygote of a mammalian zygote. Sorry, I'm drawing from all sorts of things, non-mycoplasmal, but that's the proposal, is to move on to these simple systems like a human zygote to a more complicated system like mycoplasma. And so then this is a resource. We hope to provide resources to the community. We also hope to use them. And to John's point about everybody being collaborative, this is something I was pleasantly surprised to find existed when I was looking for it, which is SYN 3.0 E. coli codon optimized version of all proteins. Not quite all the proteins, 395 out of 460 so far, last time I checked. Thanks to Keone Gondal. I think he did most of the heroic work here and then this is a list of the community itself, probably longer than this list now. I'm on that list as we can help others with this. This is free. I love the price tag here, $0, and we hope to return with interest some other community resource paying it forward. Like the antibodies, for example, that we'll make to all these native and denatured forms of all these proteins expressed in E. coli. So that's it. I'm sure I left a lot for questions if there are any. Yeah. Thank you. I'll stop sharing, I guess.
George, thank you very much. When I see this, I am just astonished by how much more we can image today than 5 or 6 years ago in terms of understanding how things fit together in a cell.
Right. I mean, I think we found time and again that actually imaging things is quite different from what you get from predicting them. I mean, eventually the two work together very. You need modeling. Even when I was a crystallographer in the 70s, and ever since, you need modeling to work hand in hand with the interpretation of the electron density, say. Essentially, all of these produce something equivalent to electron density space filling. In the case of the fluorescence microscopy, the kind of the newest entrance to high resolution, along with cryo-EM, this has the advantage over X-ray diffraction cryo-EM in that we have so many colors, so we can actually say what is it at every pixel or voxel, 3 dimensions. So yeah, we've come a long way.
So the floor is open. Don't feel the need to raise your hand. Just, dive in. With the minimal cell. So what tool do you think will likely be the most useful?
Well, being a synthetic biologist is 90% of my activity. I mean, what I've described so far is mostly analytic, but our lab as a whole, I was more inclined actually to do synthetic mycoplasma genome than a natural one, because we can change it, because it's kind of like already in the working with something that's already changed, and so to make a change on that is easier than going back to and trying to line up. But we probably will eventually line up the progenitor parental multi-omics as well. But I think that's the most exciting resource is of both the proteins to which we will make antibodies, and to all of them. So it'll be the first organism with a complete set of antibodies. But we would also excited about anybody who has a suggestion for a variation on the synthetic genome that would, let's say, help us do our job. Might put orthogonal chemistry on particular proteins, or it might change. You have one where you have really gigantic versions of mycoplasma. We're mostly motivated to keep it small biologically and large physically by expansion microscopy or by super resolution. But things like that, I think, are exciting.
I mean, I would still argue that our mycoplasma system is the most genetically malleable thing there is, in that I can change any base I want. Now you can do that with coli, but I can do this maybe a little easier than you guys can. We are starting a process to try to learn how to transplant other cells. We got some NSF funding for that. And I'll be working and talking about that, the last talk of the day. But I still see trying to figure out cell division. This may be a really terrific system to figure out aspects of cell division. That cell paper that James Pelletier and Li Ji Sun from our group put out a year ago, in that with the really minimal cell, the 3.0 as opposed to 3A, we have something that may look like cell division before the evolution of modern cell division. Similarly, we may have inadvertently, by removing the gene upstream of bacterial protein, histone-like protein, we've gone from 10,000 copies per cell to 28, or just a few copies per cell, and that may be what life looked like before the invention of chromatin.
Fascinating.
So yeah, there's some cool things that we can do, and then I gave you the tools to add anything you want to the cell, and if you need help later to take some things away, we can do that in collaboration with you.
Sounds great. Terrific resources. Thank you.
Well, I'm thrilled to have you as part of the community. You and Ed and your friends, and we'll meet again next year, if not sooner, I hope.
Well, I look forward to the rest of the talks.